Welcome, everybody, to the August edition of Chestnut Chat. Thanks for coming. This meeting is being recorded. If you put something in the chat, if you put it in the Q&A, it's going to be recorded forever. So if you hit the yes button, that means you agree to that. Thanks for coming. And Lisa.
Thanks, Sarah. Sarah and I were talking about how we used to really dread public speaking, and I never would have thought Sarah would ever be anything but clever at speaking on her feet. I mean, she did a webinar last night. She's talked to numerous media outlets in the last couple of years. So we're just glad she's such an amazing spokesman and facilitator of Chestnut Chat. I'm Lisa. I'm the president and CEO of the American Chestnut Foundation. Honored to be a part of this really ambitious mission to bring back our favorite tree. I think I can say that safely. And I'm delighted to have Ron and Greg here. I just met Ron this morning on Zoom, but known Greg for a few years now, and I think you're going to be very entertained and educated. So with that, Ron, take it away.
Sounds good. Thanks, Lisa and Sarah. Well, it's nice to be here with everybody. I'm going to give a shorter overview talk of some of the work we've been doing over the past year and a half, and then that'll hopefully prompt a lot of questions and discussion that Greg and I'll answer together or debate together. We'll see how similar our answers are. So this talk was, well, I should share my screen, was recently given at the oral session for major nut breeding programs. This is probably a generous way to put our efforts that we're developing at the American Society for Hort Science conference. And I'll just dive into it. So the center has long facilitated decentralized on-farm genetic improvement of chestnut. And so today's talk overviews a new USDA agriculture marketing service funded project to evaluate on-farm seedling families and work towards formalizing a participatory network for next generation chestnut improvement in the Eastern US. Our subtitle for the activities a part of the project period of the grant are characterizing genetic diversity and ancestry of superior on-farm germplasm. And the major people working on the project are, of course, myself and Greg, Jean Romero-Severson of the University of Notre Dame, Mike Gold, who is the Associate Director of the Center for Agroforestry, and two senior research specialists in my lab. If you're a participant, you've certainly interacted with them, Nick Meyer and Brian Weber. So a little context. This is maybe less relevant for folks attending a chestnut chat, but important to kind of set the narrative. So to date, UMCA chestnut improvement has included cultivar conservation, evaluation, and then OPC dispersal. So a cultivar collection was established in 1996 with over 60 cultivars maintained today and curation ongoing. Majority of the cultivars originate from growers or enthusiasts importations and evaluations over the 20th century, where OPC from grower selections was frequently circulated. In 1999, a subset of 12 promising cultivars was established in a replicated trial in New Franklin, Missouri, leading to the first cultivar recommendations for the Missouri River Hills, which is an ideal environment for growing, excuse me, Chinese chestnut. Because of delayed graft failure, incompatible interactions, which I'm sure many of you have seen, are kind of depicted on the slide, and interest in cultivation across a wide geographic range. Growers outside the Missouri River Hills opt to plant seedling orchards most often, which again, most of you probably know. And these, as Greg has recommended over the years, are often planted at a higher density than the final density. Maybe that's double density, so that poor performing seedlings can be culled. So to support the success of seedling orchards, the University of Missouri Center for Agroforestry has distributed open pollinated seed from the cultivar collection since 2008. So open pollinated seed distribution. Over 1,000 growers have received UMCA repository seed over the years Some of those, I think around 700 farms, are depicted on the map that you see in the blue dots and the red dots. So they either receive seed directly from the UMCA or a local nursery partner. There are some out-of-state nursery partners, Greg, for example, which account for additional growers in the eastern states, but they're not yet depicted on the map. So the point being that these materials represent an invaluable resource to select improved offspring in diverse environments to serve a number of purposes, but possibly even next generation breeding parents. So we want to build on these improvement efforts and evaluate those on-farm seedlings. So this new project overview. So last year, like I said, we were awarded a USDA Ag Marketing Service grant. And the major objectives of that grant, we're now in our second growing season, are discern on-farm selections, ancestry, and parentage. And that would be in collaboration with the Gina Romero-Severson lab. Characterize genetic diversity of on-farm selections for traits of interest. Utilize data from these first two objectives to construct core collections for conservation and breeding. And then formalize a participatory network that includes where we're doing on-farm evaluations, but extends to where we might plant future seedling progeny as a new breeding initiative to begin next generation improvement. So the point being that next generation seedling orchards could be more improved or more systematically designed than last generation seedling orchards. Okay, some assumptions and goals. The first is that seedling orchards on farm are still comprised of diverse, large families. A snapshot of 12 growers is depicted to the right, and this comes from an NGA talk, a slide that Greg gave a few years back. Altogether, there are over 30 families circulating or that have been circulated to on-farm locations, representing 3 populations. And if you estimate based on nursery records, possibly over 15,000 seedlings on farm before culling. A second assumption is that growers' observations can guide evaluations, like university evaluations, to the best on-farm trees. So the leading criterion is consistent year-to-year production, although there are other relevant criteria, including low nut defects or unique recombinants, for example, where male sterility from European or Japanese hybrids or American ancestry is discovered in individuals with high kernel quality of the Chinese chestnut. So we anticipate evaluating 5 to 8% of on-farm seedlings to identify core sets for conservation and breeding. So that means that these current on-farm evaluations that we're doing probably include somewhere between 750 to 1,200 trees. At least we're working up towards that goal. So participants, active on-farm evaluations and those scheduled for about 4 to 6 weeks from now are represented by the red dots. So together showing 30 growers across 11 states.
Hey, Ron, I'm sorry to interrupt you, but Hill had a question, I think, on your last slide about 3 populations. What do you mean by 3 populations?
So that'd be like a seed orchard or something like the Dunstan population. One seed orchard could be that's at Hark, like the peach chain core population.
Okay, thank you.
So instead of maternal families, a population or a seed orchard. Yeah. Okay. So last year, 12 growers across 5 states were included in our evaluations. We took phenotypic data and collected twig tissue for genotyping on 287 selections. The number was a bit lower because of the severe double frost that basically removed the crop from many northern environments. So this year, we expect the number to be up near 600 selections. As we move forward, we can expand our range further and offer a selection guide for selection criteria guide or phenotyping guide to growers that are in more remote environments. And then they could send us in subsamples of twig tissue and nuts to do more quantitative phenotyping should they want. Okay, so I'm going to give an overview of our genotyping and phenotyping workflow. Since last year was the first year we collected data, we won't be presenting data yet, but giving you an idea of the data that we're collecting. So again, as many of you might know, many cultivars are Castanea mellissima, the Chinese chestnut, or Chinese chestnut complex hybrids with various other Castanea species. Interspecific ancestry is common due to, or presumes common due to the dynamic sharing of OP selections over the 20th century, and then sharing resulting OP seed from those selections in sort of an ad hoc way amongst grower networks. So the Ramirez-Severson lab used a training panel, and that is the structure analysis output you see here to develop a set of 45 EST-SSR markers capable of, excuse me, I'm going to do that all talk, capable of discerning complex ancestry amongst Castanea hybrids. So you could see that the vertical bars and the colors represent relative co-ancestry of the various species. So blue represents ancestry from Chinese chestnut, while the other colors represent the respective species names that are listed below them. If the vertical bar has mixed colors, there is hybrid ancestry. Let's see. So the UMCA cultivar collection and the 287 on-farm seedlings collected in 2020 are currently being genotyped. So the resulting data will help us reconstruct not only complex ancestry, but also parentage. So we can confirm the maternal parent is accurate with the grower's records. And also if the seedling is a descendant from UMCA, which is how we're structuring the individuals that we actually incorporate into the study, we should be able to, through paternal exclusion, figure out who the pollen parent was of that offspring. So this will allow us to draw some insights on parental combining ability and potentially beneficial interspecific ancestry. Okay, so phenotyping includes 35 characters. So this is the phenotypic workflow, or at least a snapshot of what we did for the 287 individuals from 2020. There are 7 field characteristics, including characters like number of burrs per shoot, a qualitative yield estimate, and observations of male sterility. And you can see some pictures of male sterility on the upper right. The male sterile selections generally yield up to 2 times that of productive Chinese chestnut, although kernel quality can be lower. 16 nut and kernel quality parameters were considered. The most important including size, shape, color, texture, and taste, and with low variance in a given tree. Larger nut sizes are generally preferred, with a selection threshold around 10 grams per nut on average. So records are also kept on the incidence of 7 pests and diseases, some that are listed there, and new sources of resistance to gall wasp, weevil, and 2 leaf pests have already been identified. Much of that has been done in collaboration with Bob Staley, who has very high disease pressure or pest pressure in some of his fields in central Ohio. Okay, so looking forward to the utilization of these on-farm materials, we've begun working with growers, a lot of conversations with Greg to define a mutually supportive organizational structure where the circulation of resources and activities are well defined. The point of presenting this figure is to give an overall view of resource movement between the university and growers, so that past on-farm improvement can grow from an ad hoc spontaneous activity to something that is coordinated, systematic, and institutionally supported. I'll highlight a few aspects of resource movement, and note that the figures included in a perspectives paper that is in review in Frontiers, which details the breeding network in an introductory fashion and objectives in greater depth, should anybody be interested in reading that. Hopefully that'll be published later this year. So some of the bolded arrows that you see in the center are what I'll discuss in brief. So with the university on the right and the decentralized grower network on the left, you might see movement of full SIB progenies or diversity panels of sort. So like diverse maternal seedling families that are similar in composition, like volume between those maternal families. Scientific methods and guidance, genotyping, trait screening, or even marker-assisted selection at the nursery level before progenies are decentralized. Germplasm management database, which I'll talk about towards the end of the presentation. And then going from the growers to the university, the first thing might be obvious, but very valuable, and the TACF does this well. They're sort of like something to look up to in this regard, an expansive program footprint, and then observations and evaluations, preliminary selections, and selections, seed, scion, and pollen. Another important aspect, which we're encountering at HARC, is in situ conservation. So eventually, any institution is going to reach a capacity for ex situ conservation. And with the germplasm management database we're using, we could start to, the University of Missouri, integrate more in situ conservation with RAMAT tracking in that. So I'll move on from there and be happy to share the paper with anybody that's interested once it's published. So some breeding goals by class as we're looking forward. Stable production by environment is a major one. So this might include late leafing or frost avoidance. Individuals like Ching being incorporated into schemes. Early fruit maturity and leaf sinensis is definitely an aspect of production stability, particularly as we think about more northern environments. Of course, high kernel quality and low defects. I think folks might be pretty well versed with some of those aspects, but Chinese chestnut or some of the more elite Chinese chestnut cultivars being our current standard for high kernel quality, like Ching. High yield, we did mention some of these in our current phenotyping workflow, like male sterility and burrs per shoot. And then durable resistance. So with a few partners, we started actually making crosses with on-farm selections this past year to incorporate or begin to pyramid resistances to different diseases or pests into particular crossing schemes. So some of those being, well, 2 different leaf pests, gall wasp and blossom and rot resistance. So as we move forward and we're evaluating progeny on farm, emphasis will be placed both on the individual, so making clonal selections, if you will. That could be next generation breeding parents, or most importantly, in the short term, family performance. So figuring out which families actually progeny test well and produce more superior or consistently performing offspring on average compared to other progenies. Okay, so some pictures on cross-pollination. So we're initiating and scaling pollinations at the university. This wasn't something that was done before my arrival in 2019. And at present, the crossing schemes are designed to more systematically leverage the cultivar collection compared to past OP dispersal, which was basically structured based on what was requested from growers. So you can see how the pollination bags are up in the canopy, where the pistillate flowers are a bit more dense on burrs per shoot. So we use tow bucket trucks to get up there. So in 2021, pollinations are estimated to produce around 7,500 full siblings. 3,000 pollination bags were placed and pollinated by a dedicated crew. And actually another nearly 1,000 were put on farm. So the 3,000 just represents those that were made at Hark. The near-term emphasis of the crosses is to develop a large partial diallel and a variety of mapping populations segregating for pest and disease resistance. The partial diallel was made using 22 cultivars in 2 to 6 crosses each, totaling 65 crosses, and over 100 offspring are expected per cross based on the bag volume that we placed or that we used for each cross. We're currently sequencing the diallel parents, actually in collaboration with Jared Westbrook and Jason Holliday, and actually the remainder of the UMCA cultivar collection on the Illumina NovaSeq platform at 30x depth, and plan to take a multi-parent QTL mapping approach with this resulting partial systematic diallel. In the coming years, we'll begin incorporating on-farm selections into the pedigrees. Okay. So I want to briefly mention Ibrita, the germplasm management database that the project's using. Most grower records, I love this slide because it gives me a chance to poke fun at Greg a little bit, look like the sheet that Greg's holding here, which is, I believe, his original field map from the 1970s. If it's not literally that old, it certainly looks that old. But Greg helped pick out and actually led the decision on picking Ibrita. So that's why I feel like I can tease him a little bit about his field map. So Ibrita will allow us to set up standardized data entry templates within the database. And its field app, the screenshot which you see here, which can be used to call QR labels that are on the trees for direct data input and cloud-based archiving once you bring your field app back to Wi-Fi. So it should help us provide some consistency and automated quality control for on-farm data entry. And the applications here are several-fold. So first, for in situ conservation. Second, for ramette tracking. There's actually a nursery stock management function within it. And then seedling family tracking as we go from control crosses to our nursery, to on-farm locations, and set up the initial field plot. And just a little bit about the field app interface, just to give you a taste. We're actually just starting to dip our toes into using it this year. So it's new to us as well. But on the left, you can start with what would look like basically a plot list and the pedigree IDs, so the individual unique genotypes you have for that particular plot. And if you click on one, it'll take you directly to that individual. If you click on the properties tab, maybe I could pull my, if you click on the properties tab here, it'll take you to the properties, and you could toggle on and off the different phenotypes that you're taking on that day. And it should automatically populate a data entry sheet for you. Right now, you're looking at the data entry for just one ramette of peach within our cultivar repository. And then if you go back to the actual full data sheet, you could see all data entry are entered, for these spring phenological traits across each ramette within the repository. And so then again, as soon as you're back to Wi-Fi, this would automatically populate in our cloud-based database. Okay, and that's where I'll kind of turn it over for Greg and I to take some questions. But quickly, I'll say thank you to our funding agencies. So the USDA Ag Marketing Service, the University of Missouri Center for Agroforestry, and our close partner, the ARS Dale Bumpers Small Farm Research Center in Arkansas. So.
Thanks, Ron. And then Greg, did you want to say anything? Did you want to talk, or do you want to jump into questions?
I will just add a few comments, kind of background or foundational information that I'm thinking people might wonder about that Ron didn't say. And that is, first of all, we we have chosen Chinese chestnut as kind of the chestnut species, which is I'd say the foundation of the breeding program. And the reason we choose Chinese chestnut is one, it's the best adapted to the climatic range over which we're thinking about making commercial orchards. Also has blight resistance and phytophthora resistance. Kernel quality is the best of all the species. And it also is the most variable of all the species. So we got a lot of genetic variation within Chinese chestnut to work with. And I think it's also what I consider to be the progenitor species. Like all the other chestnut species are derived from Chinese chestnut. It's where it all began. However, the Chinese chestnut itself does have some limitations or defects that we need to bring in genes from other species, and maybe one of the most notable is that other species of chestnuts, in particular, I'm thinking of European Japanese and maybe Euro-Japanese hybrids. A lot of them yield quite a bit more than Chinese chestnut does. So we're thinking by bringing in some other species, including like characteristics of male sterility, we can improve the yield of, but also try to maintain essentially Chinese chestnut quality. So that we're kind of using Chinese chestnut as the foundation and bring in maybe characteristics from all the other species, but they're essentially what we call a Chinese pure or Chinese hybrid. The other thing that's, I think, kind of seems a little bit strange to me about the program is the interest in growing chestnuts commercially is a lot in the upper Midwest, Iowa, Illinois, maybe even to Wisconsin, a lot in the Northeast, upstate New York, and into New England. These areas both have colder climates and shorter growing season than most of the commercial chestnut areas in the world. And so it's an important part of this program to try and push the adaptation of the chestnuts kind of into these more northern climates for reasons that are not entirely clear to me. I think the sweet spot climate-wise for growing chestnuts in the U.S. would be in the middle South and the deep South, where the climate there matches like the commercial areas other places in the world. And I think we will expand there, but right now most of the interest seems to be from the more northern areas, which then requires a little bit more intense selection for short growing seasons and cold winter temperatures. So I think with that, I can answer questions, which I'd rather do than just keep yakking.
Excellent. Thanks, guys. Well, so let's address that first question and kind of keep going on with why are they in these locations? So Tyler asks, what is it about Illinois and Missouri that seems to make them good places for commercial chestnut operations? They seem to be very concentrated there.
Well, I think Greg would probably answer this question better, but I'll start by saying, if you're saying it seems like they're concentrated there based on the map I showed, that's probably not a good way to base your perspective on the distribution of chestnut orchards. Like I said, that represented the farms that the UMCA has sent OP seed to, or our nursery partner who is also in Missouri. So those resulting farms are proximal to the providers of seed and don't represent all farms in the Eastern US. But as far as why it's a good climate, I don't think there's a couple big things, but so the big things are, we don't experience the late frost crop loss as frequently. I think in the historical yield data at Hark, it's happened severely once since the repository has been established, around 1999, it was in 2007. I think the more north you go, you see it happen multiple times a decade. The cold hardiness is also an aspect. Greg, do you want to talk about it at greater depth?
I think the reason people want to plant chestnuts where they do has more to do with the people. It's like that's where people are who, for whatever reason, are interested in growing chestnuts, and it's almost always the case. Okay, I want to grow chestnuts. Will they grow on my site? The growing sites themselves don't say, "Hey, I want to grow chestnuts." It's got to be the people who own the land, and so it's more of a human cultural thing than a than a site selection. And I would say that over the years, I have talked more people out of growing chestnuts than I've talked into it, because a lot of people say, "Oh, I want to grow chestnuts," and I start asking about what kind of site they have, and then it's too wet or too much clay, too much limestone. A lot of reasons to talk people out of it.
So Mike Gold added in the comments like 2 points that kind of mirror what I said. Kind of substantiated our frost avoidance ability in our area. But then to build on Greg's point, like the Missouri River Hills are extremely deep soils that are well drained and well suited to chestnut, and it's quite a big region, like in and around the Missouri River. So those upland areas are pretty ideal soil texture wise, and the climate doesn't allow you to basically have the spring frost kill or the severe like winter extreme temperatures that you'd have in like Iowa.
Well, kind of along those lines, Tony asks, do you think North Alabama is a good location in the Cumberland Plateau area?
I think it's great. If you get the right soil and good air drainage or some other frost protection. So generally climate wise, it's ideal. You just got to find a good soil site.
So this is generally, most of these folks are interested in American chestnut. I like to talk about other chestnut species because I think it's important to know what else is going on out there. But some of these questions are about American chestnut specifically. So the first question, very general, what do you think about Darling 58, about the transgenic American chestnut?
I think for our use, for growing trees in an orchard, I don't think it has much value. In other words, if we were to use that, we'd have to use an American chestnut as a parent, and that would bring in all the characteristics of American that we don't want. Blight resistance, surely, that might not be in the Darling, but phytophthora resistance, small nut size, there's a whole list of things that we don't really want and don't need from American chestnut. So we have no reason to use that. Kind of genetically and also kind of socially and politically, people don't want transgenic trees for whatever reason. So I guess we don't need it is the main, answer to that.
I know one thing that has been talked about, I guess, with Chinese chestnuts, but maybe with a European hybrid bringing in, well, and I guess maybe not so much Darling 58 with an American background, but working with a disease resistance gene. And so maybe that's a better question. What about transgenics within a Chinese chestnut or commercial chestnut industry? Is there any room for incorporation of disease resistant transgenes for commercial production?
I'll go first. So, I don't think so, because it's costly to implement, and I think that we can make genetic gains with applied approaches, especially when integrating genetic tools. And we don't need to achieve the final product to deploy seedlings, which in some respects, my understanding from afar is that you guys might have to. And there are barriers, as you probably know, to deploying a transgenic tree. So basically, we can deploy seedling orchards that are commercially viable while doing genetic improvement simultaneously. So I think that we can grow the footprint of the industry and grow the applied breeding program capacity simultaneously without needing to do transgenics.
Yeah, I would just add that, basically we don't need it. We've got a lot of disease resistance and even insect resistance within our populations. And for the same time and money investment that you might invest in developing a transgenic tree, we could grow hundreds of thousands of seedlings, and they would be providing income to the growers, while developing a transgenic tree does not provide income to anybody. So there's no reason, and we have a system that we can make the genetic improvements without,
Right.
and basically have it funded by nut production from the growers. That's sort of the key to this whole program.
And I think the transgenic approach was really well suited to TACF's efforts as an alternate to multiple generations of backcrossing to get towards a more pure species or pure product, right? Pure dentata product, where having complex hybrids is not really a drawback. I'll say drawback here for the orchard production.
Good. So some more chatter here about suitability and site suitability for what you guys are doing. Someone suggests, I was wondering if it was a soil thing. My sense is that the area overlaps with black walnut growing. Can you guys address that?
I think that that's generally true. The upridge, deeper, well-drained areas are going to be fine. I mean, you do find native black walnuts in lower areas in the river hills of Missouri.
Yeah, I think the river hills can grow a lot of things that, generally black walnuts, black walnuts will grow a lot of places, but they do the best on deep, well-drained, kind of floodplain soils, whereas chestnuts are mountainside trees. And there's a lot of areas where chestnuts would do really well and black walnuts wouldn't do so well, and also vice versa. But certainly if you're in the river hills, you could grow either one just fine.
And I'll add, just from an American chestnut standpoint, caveat that I'm in Pennsylvania, usually black walnuts are on a wetter soil than American chestnuts. Like I've seen this in Indiana as well, where when you see black walnuts growing, I think Chinese chestnuts can typically handle a wetter soil than an American chestnut can. So just because and I've had this question before, if I'm growing Chinese chestnuts, can I grow American chestnuts? Sometimes. From a black walnut standpoint, I tend to say, you've got the jug loan issue. If you're growing them right next to one another, it will hinder growth, at least of American chestnuts. I don't know about Chinese. But then typically the black walnuts that I see growing in Pennsylvania, Ohio, Indiana are in wetter soils than I would wish to put an American chestnut in. They will grow. American chestnuts will grow there, but they won't grow to the best of their capacity. I've got a lot of people asking about where they can get American chestnuts, so I'm going to address that. And then Kendra said that she'll put something in the chat too. But American chestnuts, the American Chestnut Foundation does distribute American chestnut seedlings. We usually sell out in 24 hours. I'm sorry about that. We have a small seedling program where, if you're a member, we send an e-blast out. We have people, at 12:01 a.m. making their seedling purchases. So that's a tough way to get American chestnuts. A lot of our chapters, a lot of the state chapters will also distribute seeds to members. Some have separate seedling programs. So if you're a member of the local chapter, and you want to get some straight American chestnuts, there's no resistance in these things, at least very little or no resistance in these things. But all of those are available mainly as a way to get people accustomed to growing American chestnuts. And then go out and find an American chestnut. You see if they're fruiting. That's another good way to go and get nuts. There are nurseries that claim to sell American chestnuts. Present company notwithstanding, I can't guarantee the quality of seedlings from those nurseries. So y'all are welcome to buy them. People sell them whether or not they're an American chestnut or not. You guys can send us leaf samples and we can tell whether or not they are American chestnuts. But that's the best way to do it. Thomas in Maine says, is that Clack? I can't tell. It's cutting off your name. Maybe that is Tom Clack. I have 50 American chestnut seedlings in pots in Maine and wondering if anyone nearby wants some. Maine's a big state. But I guess throw the chat in there with Tom if you want to get his chestnuts. Let's see what else is in here. Okay, I'm gonna go back to the queue. Oh man, that blew up. I was at 3 questions and now I'm at 12. Okay. Where we go? Okay. All right. A couple people asking about Dunstans. We get questions about Dunstans all the time. We had one grower say, I have 2 Dunstans that are 8 years old. They're in great shape. Can you use them? And then someone else who asks, where does Dunstan fit into your program? Can you guys address maybe first tell us what Dunstan is, and then if and how that fits into your program?
Greg, why don't you answer
All
the
right.
first question?
Yeah, we, the slide that Ron showed that had all the little colored bars of the different chestnut accessions and what their species composition. It turns out that in that analysis done by Gene Romero-Severson, we did include 3 of the Dunstan chestnuts. Samples were actually sent by Bob Wallace from the ortettes of the tree, so there's no chance that they got mixed up with something else. There was basically, they were all 3 Chinese chestnuts. There was no American ancestry in them. One of them had like maybe 10% European, which was still a head scratcher where that came from. But basically, like a lot of us have thought for decades, the Dunstan chestnuts are essentially Chinese chestnuts. And, as far as how they fit into what we're doing now, I think the Dunstans are fairly good. They're not, maybe not the best we have. They seem to be better adapted to more southern regions because they ripen late in the season. And so they're not, even though they may survive the winter, they don't ripen soon enough as far north as I am in Ohio. So, and I think the Dunstans have become part of some of the cultivars that we're using. So yeah, they are a contributor to what we're doing, but not the main thing.
Okay, good. I have to brag. Oh, sorry. Did you want to add to that, Ron?
Well, the second question, I think you asked 2 questions. So one was, what are they? And 2 was, are they relevant? I think they're maybe a part of a broader genetic base. There's a lot more diversity out there than just the Dunstan population. And so our focus is using that. But there are people that have descendants from the Dunstan population on farm. And if they have a really good reason, I'll listen to it.
I just want to add one more thing, because a lot of people have planted Dunstans in a lot of places. And if you look at the kind of the online chatter about that, you'll have people that say Dunstans are great and people who say Dunstans are terrible. And mostly they haven't compared it to anything else. That's all they have is Dunstans. And so when I hear people say, oh, they're doing great or they're doing terrible, what they're really telling me is about their site. What they're saying is, I have a site that's good for chestnuts, Dunstans look good on it. Or I have a site that's terrible for chestnuts, Dunstans look terrible. So it's not the Dunstans they're telling us about, it's their site.
I think that's an important follow-up to a previous conversation on, can I grow chestnuts here or not? Like the answer is not binary. Performance at the orchard level isn't binary either. Well, some cases it could be if you have a really bad site, but usually there's a degree of quality or like degree of suitability, right?
And then I can grow an American chestnut on a poor site. I've grown Chinese chestnuts in 6 inches of soil on top of limestone bedrock. It lives. So can I grow a Chinese chestnut in that soil? Yes. Do I want to? Will it actually give me nuts? No.
Right.
And literally 6 inches for like 20 years. So it lived. But yeah, I think it's a continuum. And so shameless plug, I did a chestnut chat on site selection a couple months ago, so definitely recommend you guys check that out, and that will give you sort of the basics of what you need to know of. I go in, the tools that you can use to determine site suitability and whether or not the chestnuts will not only grow, but grow well and give you what you want to out of that planting. Let's see. Mike Alcott. Hey, Mike. Based on the understanding that all chestnut species evolved from Millissima, do you think it is possible that timber type growth habit is a recessive characteristic?
I think first of all, this idea of dominance and recessiveness is probably not even something that applies to what's undoubtedly a quantitative trait like timber type growth. So whatever you define as timber type growth, and this is actually something I've thought about and observed a lot about, but and there are components of timber type growth that have to do branch angle growth rate and ultimate size, which are somewhat independent of each other. But I think a couple relevant things is, first of all, when I have been in China and looked at trees there, a lot of the Chinese chestnuts are in fact timber trees in China, because the forest there just is not as tall as the forest in Eastern North America. So the Chinese chestnuts are adapted to growing in a forest in China. And then when they arrive here in North America, they say, wow, I can't get that tall. And so certainly the American chestnut and some of the European chestnuts and even some Japanese chestnuts do get tall enough to compete in a North American forest. And I think if we want tall trees, I think we can accomplish that through breeding, but it's a quantitative trait with a lot of components and not something that we think of as being dominant or recessive. All chestnut trees at some point in their past have been adapted to the environment where they grew. And it's not that Chinese chestnuts can't grow in a forest, it's just they grow in a Chinese forest.
Good point. All right, so I think I got Thomas's last name is, I lost it again. It starts with an L. But he also asks, is there a leaf sample kit that available to the public that shows percentage of various chestnut genetics within a tree? Because I have several crosses. Not yet. I know, I'd like to see that where we could have that available. Ron, Greg, are you guys hoping that we have something available to growers en masse, like a kit that we can send them, they send leaves back and get it genotyped, or?
I don't know about a kit, but a service would be nice as a first step towards a kit, possibly. We have talked about that. I know one of the goals, there's a layer of complexity to answering the service side of things, but one of the goals we have as a part of this project is to kind of bring the genotyping that Gene's lab does at Notre Dame over to our lab at Missouri. Now, those EST-SSR markers are very informative now, but there's a lot of full genome sequencing going on, and maybe there'd be a next-gen technology that would be cheaper and more easy to use that comes online within the next couple of years that does the same thing. So that's my short answer to that. Yeah.
Okay, good. Yeah, I agree. A service, I think that would be great. I know right now within the Chestnut Foundation, we have a tree locator program. We're trying to find chestnuts across the range, particularly on the extremes of the range. And most of what we do is morphological. So we get the leaf, we look at it, does it look like an American or whatever? And then we do pretty good on the pure species, but in the hybrids, it's virtually impossible to do it morphologically. So I'd like to see a service like that available. And I think, we might get there in the next couple of years, like you said, Ron. There's some good conversation over here in the chat. If you guys haven't had a chance to check it out, I want to point out a couple of comments that Michael Gold has pointed out that I'm guessing at Hark, is that right? Where you guys are growing walnuts for nuts, not timber. And I think that's an important distinction. We were talking about walnuts a little bit back, and I'm going to circle back around to this, but I wanted to bring up that comment and say, kind of with site selection and planting, Are you growing for timber? Are you growing for nuts? Are you growing for wildlife? These are vastly different goals that will dictate where these things can grow and achieve that goal.
Yeah, I was trying to make that point, so it's nice to reiterate it, that in the River Hills, we do have wild stands of black walnut in the low-lying areas in the floodplain, and they grow just fine. But if you're establishing an orchard with nut cultivars, we wouldn't recommend planting it there. We'd recommend planting it upridge where it drains better. Yeah.
All right. And so Matthew says, well, since we're discussing black walnuts, I was wondering if some black walnuts cause the soil to be truly toxic to surrounding plants, and if so, is it true for the California black walnut?
I don't have a definitive answer to that question.
Yep, I don't know about California black walnut, but the eastern black walnut, I do know we have grown them in close proximity to Chinese chestnuts, and there seems to be no ill effect. And I think it's pretty well established that the juglone is really toxic to some plants, but not to others. And you'll find black walnuts around here. You almost always find bluegrass and raspberries growing under them, because those are two plants that seem to be just fine. And then they love the selective herbicide of the black walnuts to help them out.
Mark asks, any thoughts about future market trends? What about chestnuts versus hazelnuts versus walnuts?
I think there's a lot of lofty projections being put out there nowadays. And I think that's all that they are. So I'll say that. I think that there is growth. I think it'll probably continue in a more stepwise linear way than displacing huge import supplies, which I think that's the goal in some cases, or for some camps. Having lofty goals can be good if you shoot for the stars and land amongst what's the phrase, beneath the stars somewhere. Sometimes if you have lofty goals, even if you don't reach them, you could achieve a lot. So that might be good. I think before you establish plots, what it comes down to is you want to have a good idea of where you plan to take that market, or take that crop to market, even if it is a few years down the road, and you want to be kind of realistic with the price point you think you're going to get, and the ability of that target market to absorb your supply. The issue is really a factor of how big of an orchard you're planning to plant, and your awareness of other orchards being established around you. So hopefully you understand the distinctions that I'm trying to communicate there. But
yeah, I think at the moment the demand for chestnuts is huge, and people really like the Chinese chestnuts. And most of our customers they don't speak English as their first language. They're recent immigrants from Korea, China, and like Bosnia or other former Yugoslavian countries. And so these people, they eat chestnuts. They buy them like 50 or 100 pounds at a time. And it's what we sell out every year, way before we get done harvesting. So for us, and for all the other chestnut growers that I know of, it's just a matter of getting them out of the orchard and filling order as quickly as you can get them in the bag. I don't know how long that's going to last. There's at some point the market's going to be saturated, but it's that overwhelming demand that is really driving the interest in planting chestnut orchards. And then at some point when that market runs out, we'll have to be like everybody else and actually sell them.
Well, what are people? I'm just curious. Those communities who are buying them, are they eating them? Are they reselling them? Are they cooking with them? Are they eating them raw? I mean, do you have any indication of how people are using chestnuts in the communities that you're selling them to?
Oh, I have asked, and I've had customers for decades, and mostly the Koreans and the Chinese, and I'm not. I think the Bosnians too. They just mostly roast them and eat them, and it's a little bit like, we in North America eat potatoes. I mean, they can sit down and eat a lot in one sitting, although it's more of a snack than it is a component of a meal. And it's also very seasonal. This is the thing that the New Zealanders found out, is that the people who eat chestnuts want to eat them in the fall and the winter. They don't really want them in the warm year. It's a cool weather treat. And so, but because it's a high carbohydrate, has a composition more like grain or potato, it is something that people can eat them, by the pound and not just by the handful.
I want to come back to that point, but I've been remiss in answering poor John's question. He asked this before we even started. What are you all using for fencing?
For deer fencing?
It says fencing. I imagine deer fencing.
Yeah, that's the only reason we need a fence is for deer. And we have, the kind of the standard is 8-foot high woven wire. And I think that's certainly cost effective. I just put up a deer fence of 7.5-foot high plastic netting, which I attached to surrounding trees because I didn't want to cut them down and put in posts. So far that's working. And I think so between woven wire and plastic netting and around the orchard rather than around the trees works.
Ron, are you guys fenced in?
We do not have a perimeter fence around park. We have deer in our plots, and there could be a lot of deer in our plots. I mean, it's like the River Hills are also a deer hunting haven. They're the big cultural thing. So we probably lose some crop due to deer. There are some local growers that have the dream deer protection. They're the 8-10 foot tall woven fence. In some of our new plantings, we plan to establish just with T-posts and single wire strands, maybe 3 or 4 up to 8 feet, and then put one wire a few feet removed on the outside of electric around it, maybe 3 foot high. And that usually keeps deer out of the horticultural trials at Hark. We haven't tried that yet ourselves, but that's our cost-effective way to protect seedling plots. And if we're planting on a smaller, it's like if we were doing several acres or more. If we were doing smaller plantings, we would use tree cages.
Yeah, so Mike Alcott just says that he's successfully used 6-foot 2x4 welded wire and never had deer jump that fence. Mike, I'm assuming that's in north central PA and not New Jersey. In New Jersey, I think you need 10-foot high deer fence. Yeah. In most of our plots in Pennsylvania and New Jersey, we tend to use the woven wire, that the big, that especially if we have a plot that we want to last a long time. Even if we're not using the trees for a long time, if we want to use that plot for a long time, we tend to go woven wire. If it's a more temporary plot or something that we don't mind removing the fence in about 10 or 15 years, we use plastic. There's a lot of options. I tend to prefer perimeter fencing. It tends to be cheaper than per tree, but that depends on the density that you're planting at and how many trees and what kind of environment you have. And, like Greg said, you could save a lot of money using the trees that are already existing rather than buying posts and things like that. So there's a lot of options that you have for keeping the deer out. Let's go to Tony. He says, do you shake chestnut trees like we do for pecans and then harvest them?
We have, well, I guess on an experimental basis, you can shake chestnuts. However, they add like half of their dry weight in the last 2 weeks before they fall off. So if you shake them too soon, the nuts aren't mature and then they lose quality and yield. And if you wait too long, then they're already falling. And so trying to find that perfect time to shake the tree, it might not even exist because the nuts on one tree drop over a period of time. And so pretty much everywhere in the world where chestnuts are grown commercially, they just let them fall naturally. And then one way or another, sweep them up or pick them up off the ground. And I think that's going to be what we do.
I've had a couple questions about seasonality of trees. So Doug, oh, and I want to correct myself. So Mike corrects me. He says 30 years of growing a vegetable garden in New Jersey. He has not had deer incursions within his 6-foot fence. So I'm happy to hear that. Let's go to seasonality. So Doug Gillis says, I ate roasted European chestnuts at the Trevi Fountain in Rome in April. And this is to someone else, Mr. Sims. He asked sort of the same question. What is the best way to keep harvested Chinese chestnuts longer to maintain high edible quality? And then Doug says, how can you keep them until April like he did in Rome?
Yeah, keeping chestnuts for a long time is a challenge. The Chinese claim they can keep them until May. And the trick is, one, to keep them in cold storage and high humidity. Somehow you got to maintain a high level of moisture or they will spoil. It's, contrary to what you might think. And I like to say it's like trying to keep bare root nursery stock or carrots. You got to keep the moisture in them. And then the challenge is for keeping them, say, beyond Christmas time, is that if you got them in high humidity with a lot of moisture, they start to germinate. And so keeping them fresh without spoiling, but at the same time dry enough so they don't germinate, is a real critical moisture level. And that's the key. And it's not easy to do.
So I'll jump in and add that we just analyzed some spoilage data from a replicated study using nuts harvested from different grafted cultivars at Harp. And there's definitely different levels of exterior spoilage incidence and interior, so outside and inside the shell, by cultivar, with Mossbarger and Ching being the best as far as resisting incidence, up to 200 days. So we probably should replicate the study, but it was pretty statistically robust with 4 reps and 16 nuts per time point and cultivar. There was a number of different time points, 14. And it starkly showed that Mossbarger and Ching seemed to have a higher resistance level to spoilage molds, which is interesting because Ching is a candidate for blossom end rot resistance from Amy Miller's work. If I'm right, correct me if I'm wrong, Greg.
That's right.
So maybe there are some innate host resistances that help in both categories in that regard.
Yep. Yeah, Ron just brought up a good point, and that is spoilage in storage is a huge problem for anybody growing chestnuts. And so a large part of the breeding program is to select chestnut materials that do hold up well in storage. And it seems to be the underlying contributor, or at least one of the underlying contributors to good storage life, are chestnuts that dry slowly. So the slower it dries, the better it keeps. But this stands in kind of opposition to the idea that a lot of people have where they want to make chestnut flour. And if you want to make chestnut flour, you want a cultivar or a material that dries quickly. And so as we've been selecting for nuts that keep well, and that's been going on for decades, we are inadvertently selecting for ones that dry slowly. And so they're therefore difficult to make into flour. So if we want to go into the flour business, we're going to have to reverse kind of what we've been selecting for.
One other thing, and then we can move on. So I know the question was, how to keep nuts longer in storage. But if we want to just back it up to a general takeaway from our experiment, it was that regardless of what cultivar, you'll have minimal amounts of incidence up to like 60 days, which is the typical timeline for selling nuts right now with the current supply. Usually, the growers I talk to, Greg, again, correct me if I'm wrong, usually have sold their crop by the holiday season. So it's not really an issue at this time, but it could be a growing pain experienced if the supply rapidly increased. And so we're mindful of that with some of the parents we're using. Is that all fair? Can you agree with Greg?
Yeah. And the other little caveat I would add that people don't understand, and that is when chestnuts first come off of the tree, they're not that great to eat. The sugar content is fairly low and they don't taste very good. I think they start to taste good about 2 weeks off the tree. And so from 2 weeks to maybe a month or two, that's the really sweet spot to eat chestnuts. And if you want to keep them longer than that, you basically got to slow down the progress of what they do. But right off the tree, they're not that great. And so we got customers who want them right away. And they've been disappointed when we shipped them, within a week of off the tree. And so we just say they're not going to be ready till October 1st, even though we've harvested them in September, because they got to hit that 2 week off the tree to develop some sweetness and be good to eat.
All right, good conversation. I inadvertently missed this question. John asks, with all the rain in Ohio this summer, Pennsylvania too, I planted some Americans and some Chinese this spring and only lost one. Has the rain been a problem?
If you got a good chestnut site, you can't get too much rain on it because it all runs off or soaks through.
Sorry, where is this question? I was going to read it again.
Oh, sorry. It's in an answered at the end of the answered area. And then I guess just the question, has the rain been a problem? I know it has been a problem when we plant nuts. When we put nuts in the ground, even on a good site, sometimes they get waterlogged and die and rot before they come up. That's for planting nuts in the spring. If I plant seedlings in the spring, I want a lot of water for them to get set in. So that's for bare roots, containerized too to some degree. So, I mean, I think it depends on exactly what you're growing and where your site is and a lot of that stuff. But I generally prefer more rain than not enough rain.
Yeah, I'd say growing trees in pots, and certainly when trees are very young, it's easy to overwater them. I say there's two ways to kill a chestnut, either too much water or not enough water.
I steal that, Greg. That's one of my favorite things to say because it's true. My other favorite is, and because Hill's on this call, my other favorite thing is people ask me all the time, why did my tree die? Or why is my tree doing this? Or why is something doing that? And Hill's answer is for no good reason. And that's another one. All right, we got about 20 minutes left. Are you guys good to keep going for a few more minutes before 1 o'clock? Okay. Charles asks, what mix of fertilizer is best for small seedlings?
That's one of those, it depends. And, on soils around here where we got like no measurable nitrogen, it's got to have high nitrogen. But it sort of depends on what the tree needs. And I think chestnut trees do respond to fertilizer. They seem to be able to make good use of what I would call heavy fertilizing, but we never fertilize as much as people put on a cornfield.
Okay. And before, I want to make sure there's several questions here. We're getting back to the American chestnut side of things. So I've got a couple questions. Do you work to preserve any pure American chestnut cultivars? Does having a non-native chestnut orchard in proximity to a research center for native chestnuts affect that research? And then there was another one coming back to the OXO gene. So do we know if Chinese chestnuts have an oxalic oxidase gene, and do Chinese chestnuts vary in blight resistance? So this is all kind of coming back to TACF's work. So let's do that last one first. Does Chinese chestnut have something akin to an OXO gene, and does Chinese chestnut vary in blight resistance?
I would say, first of all, I don't think we have any evidence that there's an OXO gene in Chinese. Nobody's looked for it, but I don't think it exists. And certainly Chinese chestnuts vary quite a bit in blight resistance. And this comes as a surprise to a lot of people when they see Chinese chestnuts dying from blight or getting serious blight cankers. They do vary in their resistance. And, I think that's worth noting and studying. What was the other question?
All right, we got a couple more. Do you guys do any work with preservation of American chestnuts, or have any interest in American chestnut cultivars?
I think not near as much as the American Chestnut Foundation. I would say, we produce like 100,000 pounds of chestnuts per year. If I had 100,000 pounds of American chestnuts, I couldn't sell them because they're too small. So it's not something that, even if we had them, they'd be great. Certainly they've got really good flavor. They peel well. There are some characteristics of American chestnut that are desirable, but we can also find those characteristics in Chinese or other species.
And so Jim Tate asks, so as a nonprofit organization, TACF, interested in the conservation of a native chestnut, is growing hybrids or Chinese chestnuts commercially consistent with conservation? And I've got a couple more of these questions. How does Chinese chestnuts and these hybrids, how does that affect conservation of American chestnuts? Where is that intersection or conflict? I mean, is there a conflict? Is there a crossover? What can we learn from each other?
Well, I think maybe what somebody was asking, and I think this is part what you asked before, and that is, what happens if you have a Chinese chestnut orchard, say, adjacent to a forest that has American chestnuts in it? It's real. All chestnuts are cross-compatible, so it's pretty likely that pollen from the American chestnuts might land on the Chinese. That's of no consequence if you're just going to eat them. By the same token, it's likely that pollen from chestnut orchards, whatever is in that orchard, could blow on to nearby American chestnuts, if there were flowering ones in the forest. What's the consequences of that? I don't know. There aren't many cases in the world where we grow a cultivated crop next to a wild plant that's the same species. Has happened with apples in Kazakhstan. And I think in China, the Chinese chestnut has been for a long time cultivated adjacent to kind of the wild stands. So I think that whatever humans have been selecting for in their orchards, those genes get enriched and will impact kind of the genetics of the wild trees. So I think in cases where you got a lot of cultivation adjacent to a forest, certainly what you're cultivating can have an impact on the evolution of the trees in the forest. But on the other hand, it's so slow. Generations of chestnuts turn over, 30 years to 100 years. And so the impact of cultivating chestnuts next to an orchard or next to a forest, it's going to be something that take you 500 years to measure.
Yeah, and I mean, Chinese chestnuts have been imported, Japanese chestnuts first, European chestnuts, although they're susceptible to the blight, but I mean, Chinese chestnuts. Craig, do you have any sense of how many Chinese chestnuts have been imported and then subsequently planted across the generation? I mean, we're certainly in the millions of Chinese chestnuts that have been planted across the US, right?
Yeah, I would say we're in the millions, but maybe not the 10 millions. So compared to how many American chestnuts were in the forest before the blight, we're not close to that yet. And I would like to add, Gene found that some of those what were thought were pure American chestnuts from Maine turned out to be part European.
We knew they were part European. We knew they were morphologically, we knew they were part European.
Okay, so you were good at that.
There was hope from some people that they might be American, that we would be wrong, but the genetics showed otherwise. No, that's not. There are surprises, but that one in Maine wasn't a surprise.
And I don't know the history of those, but it's possible that those arose from somebody planting some European chestnuts, which spontaneously hybridized with native chestnuts. And I know that when the state of West Virginia collected, their remnants of American chestnuts to put in the nursery by Parkersburg, they got a lot from the Eastern Panhandle, which also look like they're part European. So I think this idea that planted trees can impact the native trees. You know, I think there's some evidence that has happened.
I think Europeans more so than others. I think, well, and it has to do a lot with how genetically close they are too. So the Europeans are more closely related to the Americans than the Japanese than the Chinese.
Well, also the American-European hybrid is a vigorous tall tree which can compete in the forest and maybe even outcompete pure American, whereas the American-Chinese hybrid might not do so well in a forest.
Yeah. All right. We're running out of time. I'm going to take one more. Oh, and Jim, yes, we can continue that conversation. And I think that's an interesting question. So his question was the consistency of talking about other species. I think, Greg, Ron, we work a lot with other nut growing organizations. I'll shamelessly plug the Chestnut Growers Association of America and the Northern Nut Growers Association of America and your local state-run nut growing association. There's lots of great people out there growing nuts of all kinds and for different reasons. And the American Chestnut Foundation tends to have a very specific reason to want to restore that species to the wild. And that's a different goal, but I don't think is in direct conflict with commercial industries.
Given more time, I think there are some good answers towards that end for Jim. Particularly from the outputs on the breeding side, not necessarily the cultivation side. But because I think you just clicked the question to answer, so I can't see it, but that's fine. You're not just TACF isn't just interested in the conservation of, but the restoration of is what they're interested in, and that does require breeding with material that isn't dentata. So there could definitely be overlap in some of the outputs from breeding cultivars or seedling populations for commercial production that do help support or diversify sources of resistance for restoration applications.
Or some of these other characteristics that are going to impact us all, like some of these cold hardiness issues or things like that. I mean, if we can figure out what the underlying mechanisms of these things are, if it's not in American, we can find it perhaps more easily in these other species and then apply that knowledge base to American chestnut. So I think there's definitely utility in studying.
The benefits are reciprocal in that way, actually. It does work from dentata over to us and the Chinese chestnut as well. And I've talked to Jared and Jason about that too, especially because of the interesting their work looking at like local adaptations and basically genome-wide by environmental associations. I find that extremely interesting, and it could really play into trying to breed populations in more of the extreme environments where we might find out later we don't have a diverse enough genetic base for it. Anyhow.
One quick addition. Some people are interested, and I've become really interested in this lately, and that is growing an orchard tree as a dual purpose tree. In other words, plant trees in an orchard that make a saw log at their base. And so I think that is where there's some overlap between what TACF is doing and what we might be doing. And the reason why I'm suddenly interested in that is now I'm in the process of got some 30 or 40-year-old orchard trees that I want to get rid of. It's really expensive to get rid of those trees. And it would be nice if I could just call up the local logger and say, hey, come take out my trees, and they would do and pay me money to do it. So yeah, I think there is certainly some areas of overlap in the breeding between what we want for an orchard and what people want to restore the forest with.
There are, and I'm going to apologize, there are more questions than we have time to deal with, especially Jim, because his question directly relates to what you just said, but Hill was in there first. And I think this is going to take some time. He said, can you talk a little bit about your Chinquapin work?
I will try to do that in the most succinct way possible. So we have some, so we're not all about we do some conservation too, I guess. So there is a good transition. The Missouri Department for Conservation provides us some annual support to collect Ozarkensis part of Missouri, where there are remnant populations that are in that suckering and dieback cycle. Sometimes they make it a little bit longer. Sometimes they're pretty stunted and putting up a lot of shoots because they're heavy with blight. But we're going and basically taking like passport information on those remnant plants in various sites in southern Missouri. We haven't gone over the border yet to Arkansas, but there's interest in doing that. And collecting leaf tissue. The project will move incrementally towards the following. Basically, genotyping, likely with a genotyping by sequencing approach, those individuals to develop markers and just do a general diversity study of those materials. So we could look at like allelic diversity or structural diversity of those populations and try to work towards identifying representative sets that can be conserved ex situ at HARC and maybe maintained longer in a more healthy state to get to flowering. And then over time, that effort will expand gradually to include more samples, try to introduce ones that actually add new variation to the ex situ core collection, and so on. We're doing many other things. So this is something that we're kind of doing in parallel as opportunity allows. We want to work towards having a nice diverse representation of what's left of Ozarkensis in wild populations and start to integrate sources of blight resistance into subsets of that collection. So if there is genetic structure, we would like to subdivide those as like the recurrent parent groups in initiating a backcrossing scheme. So that if we do try to implement some kind of program like the TACF does with Dentata, we're starting with a nice genetic base representative of what is out there now, and we're starting to integrate different sources of resistance into the spectrum of that genetic base represented in the core set. Hopefully that's sufficient. Hill, if you have other questions, I'd be happy to think aloud with you over the phone about what we have planned and should continue to plan and how we might pivot the plan. Because our capacity is stretched across a number of different crop species, as you know.
I just want to throw in another quick thought about the other direction. Chinkapins do not get blossom end rot. They have really good flavor and they have some gall wasp resistance. So there is interest in using chinkapins for breeding, bringing in those characteristics to our orchard trees.
Great. Well, I wish we had time. We still have a couple questions that are left, but I know Kendra's typing, and Jim, I'll type too, and maybe we can get Greg to answer that offline. Ron, Greg, thank you guys so much for joining us. I really appreciate your time and expertise and sharing that with our audience. Again, and I've had a couple people talk about the wood quality stuff we're going to talk about next time. So September 17th, join us, same time, same. Well, not the same link because it'll be a different link for Chuck Ray talking about wood quality. But thanks, guys, and I'm sure our paths will cross again in the future, and I will look forward to it.
Yeah, most definitely. Thanks for having us.
Yeah, thank you.
You guys.
We'll see you. Bye.